Perfect. Thanks so much. And thank you for the invitation for Hannah to present today. So Hannah Wilkins is a fourth-year PhD candidate and Institute for the Basic Biomedical Sciences Director's Fellow at Johns Hopkins University in the Biochemistry, Cellular, and Molecular Biology PhD program. Prior to her PhD, she graduated from the University of North Carolina at Chapel Hill in 2021 as a Distinguished Chancellor of Science Scholar, where she studied biomaterials for therapeutic intervention of cystic fibrosis. At Hopkins, she's completing her thesis work in the Department of Pharmacology and Molecular Sciences in my laboratory, and she's co-mentored by Dr. Ben Orsburn, where she is studying antiretroviral drug metabolism and transport in the brain and at the blood-brain barrier for the treatment of HIV. She recently completed a summer internship at Merck and became interested in transitioning to industry after graduate school. She's just about finished up with her thesis, and we're incredibly proud of her and excited she could present her work here today. So Hannah, please.
Yeah, thank you, Deanna, for that wonderful introduction, and thank you all for the wonderful opportunity to present today. The culmination of my thesis work thus far as a fourth year in the School of Medicine, which has been studying how the blood-brain barrier may demonstrate capability to influence tenofovir and M-tricytidine metabolism and transport in the brain. And so today I'll be walking through some exciting data that we recently just published last week, where I'll start first by introducing tenofovir and M-tricytidine as major antiretroviral drugs used for the treatment and prevention of HIV and their pharmacological activation pathways. I'll then show how we are able to quantify these drugs as well as their pharmacologically active metabolites in both brain and blood-brain barrier cells, and how we're able to characterize the metabolizing enzymes and transporters that may be responsible for these results that we observe, as well as the activities of some of these metabolizing enzymes and transporters. And together, how the tenofovir and M-tricytidine metabolizing enzyme transporter proteins may be further regulated in the context of HIV infection and therapeutic intervention, and may also impact other proteins as part of other cell pathways, in addition to these important pharmacological proteins. And so to begin, we know that the blood-brain barrier, denoted as BBB, prevents neurotoxic components and pathogens from entering the brain, while also allowing for the selective efflux and influx of molecules from entering and exiting the brain. This regulation is absolutely essential for proper neuronal functionality and is enforced by the unique composition of the blood-brain barrier, which is comprised of three major cell types: astrocytes, pericytes, and brain microvascular endothelial cells. Between these cells, there are also junctional proteins, such as tight junctions, which create an effective seal necessary to prevent a leaky blood-brain barrier. Due to this composition, it is quite difficult for many drugs, as we know, to pass through the blood-brain barrier and penetrate the brain. In the context of HIV, or human immunodeficiency virus, which, as we know, infects the brain and creates reservoirs of persistent virus within the brain, it is due to the difficulty of antiretroviral drugs used to treat HIV from passing through the blood-brain barrier. As a result, in many patients infected by HIV, this manifests as significant neurologic effects that may contribute to, for example, major depressive disorder, HIV-associated dementia, as well as impairments in energy and lipid metabolism, as well as immune regulation. Now, walking through the pharmacology of both tenofovir and then M-tricytidine, but beginning with tenofovir. Tenofovir has been used extensively in pre-exposure prophylaxis, but also first-line antiretroviral regimens that are given in the clinic, typically. And the way that this drug works is through pathways of what's called pharmacological activation. So it's typically given in the form of a prodrug, either as tenofovir disoproxyl fumarate or tenofovir alafenamide. It then undergoes cleavage to become tenofovir once taken. And then through nucleotide metabolizing kinases, many of which are listed here, pharmacological activation through phosphorylation is required to the active metabolite form, which is tenofovir diphosphate, or TFVDP, which competitively inhibits HIV reverse transcriptase, thus blocking HIV life cycle. Emtricitabine is a very close sister, if you will, to tenofovir in that it also requires pharmacological activation through phosphorylation. Unlike tenofovir, it actually requires 3 separate phosphorylation steps, first starting from emtricitabine to then monophosphate, then to diphosphate, and then finally triphosphate, which is the active metabolite of emtricitabine, which then, like tenofovir, competitively inhibits HIV reverse transcriptase, thus blocking the HIV life cycle. And this phosphorylation is done by a host of different nucleotide metabolizing kinases as well. And so together, we know that tenofovir and emtricitabine, through pharmacological activation, through phosphorylation done by a host of different nucleotide metabolizing kinases, as I mentioned, inhibits HIV reverse transcriptase, thus blocking HIV. And we know that for both tenofovir and emtricitabine, there are several efflux and influx transporters that are substrates, or that these drugs are substrates of, that are required for the transport and ultimately the distribution of these drugs across tissues. However, despite that, we know how the transport and metabolism works in terms of the players that are involved. We don't understand the capabilities of the blood-brain barrier to transport and metabolize tenofovir and emtricitabine. And specifically, because we know that the blood-brain barrier is actually quite heterogeneous in cell composition across the brain, we don't understand the capabilities of blood-brain barrier cells to differentially transport and metabolize these drugs, which is important to consider as we know that the blood-brain barrier, as I mentioned, is a major challenge for the penetration of these drugs and availability in the brain, thus targeting the persistent reservoirs of HIV within the brain. And so to begin to investigate this question, we first wanted to quantify tenofovir and emtricitabine, as well as their pharmacologically active metabolites in both the brain and blood-brain barrier cells, which has been done to an extent within literature. And to do this, we used rhesus macaques, which are used extensively as a wonderful model system to study HIV, specifically using SIV, or simian immunodeficiency virus. And to begin to answer this question of antiretroviral quantification within the brain and within blood-brain barrier cells, we used 2 separate rhesus macaque cohorts. The first on the top was using a historical rhesus macaque cohort from samples that had previously been banked in the past and that we had available to us. These rhesus macaques were inoculated with SIV, which again models HIV, and then they received a antiretroviral regimen, including tenofovir and emtricitabine, administered subcutaneously for 6 months, after which these macaques were sacrificed. And we were able to collect and analyze 3 separate brain tissues: the thalamus, frontal cortex, and cerebellum. And the second rhesus macaque cohort is similar but slightly different in that these were healthy rhesus macaques, so not infected with SIV, but healthy, but received a very similar antiretroviral regimen, including tenofovir and emtricitabine, through an intramuscular administration for 6 months, after which these animals were sacrificed. And thus far, we have isolated and analyzed the astrocytes from the brain to begin to answer this question. And so first focusing on the first cohort, so those that were SIV infected and receiving ART, looking at the 3 brain regions isolated, we used mass spectrometry to quantify both the parent tenofovir and emtricitabine drug on the left, as well as their pharmacologically active metabolites, tenofovir diphosphate and emtricitabine triphosphate on the right. Here you can appreciate the concentrations of tenofovir and emtricitabine and their active metabolites across the 3 brain regions: the thalamus, frontal cortex, and cerebellum. Here we do have to acknowledge that we were limited in sample availability. Again, as I mentioned, this is a historical rhesus macaque cohort. However, we have seen extensively within literature that both tenofovir and emtricitabine can be quantified within the brain. But also, too, we're able to quantify their pharmacological active metabolites, which again is what's responsible for inhibiting HIV. And we can appreciate that we are able to quantify quite heterogeneous measurements of both parent and active metabolite across brain region. Particularly, we're able to quantify significantly more tenofovir and tenofovir diphosphate relative to M-tricytidine and M-tricytidine triphosphate. And this data together confirms that we're able to quantify both the parent but also active metabolite within the brain and diversity in measurements across brain regions, which then turned our attention to looking within astrocyte cell types, which are prevalent within the brain, but also are an essential component of the blood-brain barrier. And so here we analyze astrocytes, again, isolated from our second rhesus macaque cohort, which are healthy macaques receiving antiretroviral drugs. And using mass spectrometry, we're able to quantify the active metabolite tenofovir diphosphate within the intracellular components of astrocytes, which is quite exciting to us because, to our knowledge, this had not been looked at previously within the literature. We were not able to quantify M-tricytidine triphosphate within astrocytes, which is interesting considering that we were able to quantify this metabolite, certainly less than tenofovir, but still able to quantify within tissue. Previously, groups have quantified that blood-brain barrier cells, including astrocytes, but also parasites and endothelial cells, were capable of accumulating tenofovir and M-tricytidine, but they did not analyze, as I mentioned, the active metabolite. And so we're quite excited to see the accumulation and possible metabolism of these forms of the drug within astrocytes. And this, of course, turned our attention to looking at other blood-brain barrier cells in addition to astrocytes, so the parasites and endothelial cells, to investigate whether they could also accumulate tenofovir and M-tricytidine and potentially metabolize them to their active forms. And so to look at this, we use an in vitro assay where we used human primary cells, including endothelial cells, parasites, and astrocytes, and exposed these cells to a very similar concentration or antiretroviral regimen, including tenofovir and M-tricytidine as our rhesus macaques. And we exposed these cells for 24 hours and then analyzed the intracellular metabolite formation by mass spectrometry. And here on the right, you can appreciate the concentration of the active metabolite, also known as the pharmacological activation that occurred within these cells. And here we were able to measure that all three blood-brain barrier cell types were able to convert tenofovir and M-tricytidine to their active forms, tenofovir diphosphate and M-tricytidine triphosphate in vitro. But even more interestingly, we also observed that there was significant cell type differences in terms of pharmacological activation of tenofovir and M-tricytidine, where, for example, with parasites, M-tricytidine triphosphate accumulation was observed to be about over 600% higher than tenofovir diphosphate. Also, the metabolite formation relative to endothelial cells and astrocytes as well. So we see significant differences in pharmacological activation within blood-brain barrier cells, but even more excitingly, we're able to actually see that these cells were capable of formation of the active metabolites. We then wanted to investigate what could contribute to this differential pharmacological activation within our blood-brain barrier cells. And so we wanted to characterize the tenofovir and M-tricytidine metabolizing enzymes as well as transporters that may be responsible for the results that we observed. And so to do this, we used proteomics by mass spectrometry to quantify the tenofovir and M-tricytidine metabolizing enzymes and transporters, where we use primary human blood-brain barrier cells, again, including astrocytes, parasites, and endothelial cells. We also used primary human hepatocytes from the liver to use as a relative comparison when examining protein concentrations, as we typically consider hepatocytes in the liver as our gold standard in considering nucleotide metabolism and transport. And so what we did was we lysed these cells and then we processed for proteomics by reducing and alkylating the proteins within the cell lysate, trapping the proteins, cleaning them up a little bit, and digesting them using trypsin, which of course creates peptides, which we then quantified and injected into a mass spectrometer, where we ran what's called data independent acquisition or DIA-based proteomics, which essentially scans across the mass to charge ratio that mass spectrometer measures that creates an in-depth snapshot of the proteome so we can analyze thousands of proteins per sample at a given time, which creates a very in-depth encapsulation of the proteomes of these cell types. And so when we get this data, we conduct a library search to map the peptides back to the proteins and from the sample. And we're able to quantify both the relative abundance of each protein, but also using what's called the proteomic ruler, we're able to estimate the concentrations of each protein. And from this workflow, we're first able to quantify several efflux and influx transporters for tenofovir and emtricitabine. Here we use proteomics to measure the concentration on the y-axis for ENT1, which is an influx transporter, as well as MRP4 and MRP1, which are efflux transporters. And we can appreciate here that in the case of ENT1 and MRP1, that we are able to measure differential concentrations between blood-brain barrier cell type, which is interesting to consider because this may lead to differential accumulation of tenofovir and emtricitabine within the cell. Of course, here we use Western blot just to confirm protein abundance. We also, as I mentioned, use hepatocytes as a relative comparison. Unfortunately, we are not able to quantify, using proteomics, the concentrations of these transporters within hepatocytes, which according to literature, these transporters are variably expressed in hepatocytes between patient donors, and which may contribute to why we did not see that here within our proteomics data. In addition to transporters, we're able to quantify several tenofovir metabolizing enzymes. So these 4 metabolizing enzymes, again measuring the concentration on the y-axis, are responsible for the activation of tenofovir to inhibit HIV. And just like for the transporters, we can also appreciate that we are able to measure differential concentrations between blood-brain barrier cell types. So for example, for pyruvate kinase, or PKM on the right, there's a 77% difference between the concentration of PKM or pyruvate kinase muscle type within endothelial cells relative to parasites. And so additionally, we are able to measure the second protein, creatine kinase brain type, or CKB, which is a very important protein specifically for tenofovir in that it is responsible for that last activation step to the active metabolite, particularly within brain. And here we know from literature that astrocytes extensively express CKB, which is what our proteomics here shows as well, but also actually able to quantify those concentrations relative to the other blood-brain barrier cell types. Again, this data suggests that similar to the transport, that the differential concentrations of these metabolizing enzymes could lead to differential metabolism within blood-brain barrier cells. And similar to the tenofovir metabolizing enzymes, we're also able to measure differential concentrations of emtricitabine metabolizing enzymes. So again, here are all the emtricitabine metabolizing enzymes required for the activation of emtricitabine to its active form. And just like the transporters and tenofovir metabolizing enzymes, we're able to quantify heterogeneous concentrations between blood-brain barrier cell types. And just like with tenofovir as well, what we can appreciate is that for several of these proteins, they're actually quite comparable or even higher in concentration to hepatocytes, which is interesting considered that we typically think of the liver as a major metabolizing organ. But here within blood-brain barrier cells, we can see for many of these proteins that they're comparable or even higher in concentration to our hepatocyte measurements. We then wanted to complete this picture of the characterization of not only looking at concentrations of these proteins, but also their activities. So whether or not the metabolizing enzymes and transporters are active and maybe even differentially active between barrier cells. And so to first begin to investigate this question, we wanted to begin to look at our efflux transporters. Transport proteins responsible of effluxing substrate out. Using an efflux transporter assay by fluorescence activated cell sorting, or FACS, to measure the activity of these efflux transporters. And the way that this assay worked is that we plated, again, our primary human blood-brain barrier cells, astrocytes, parasites, and endothelial cells, and we exposed them to fluorescent substrate for 3 different efflux transporters for either tenofovir, prodrug form of tenofovir, or emtricitabine, which is PGP, BCRP, and MRP4. And after exposure to the fluorescent substrate for these 3 transporter proteins. We allowed the uptake of these substrates into the cells. And then we measured the efflux of these substrates over the course of 2 hours, which we measured by fluorescence activated cell sorting, or FACS, which gave us the efflux capacity for all three of these proteins. And interestingly, we're able to find that all three transporters are active between blood-brain barrier cells. But interestingly, for PGP and MRP4, we saw differential efflux capacity or activity between blood-brain barrier cells. Specifically for parasites, for PGP, the efflux transporter PGP, we saw that the efflux capacity was the highest. However, for parasites for MRP4, the efflux capacity was the lowest. And so this efflux capacity, what appears to be blood-brain barrier cell type dependent. But interestingly, for BCRP on the far right, we didn't see any significant differences in terms of efflux activity or activity between blood-brain barrier cells. So it's important to consider the transport within these cell types may not be homogeneous across cell type for tenofovir and tricytidine. And so then we wanted to look at the activity of metabolizing enzymes, first starting with tenofovir metabolizing enzymes, specifically creatine kinase brain type, or CKB, which I mentioned is highly expressed within astrocytes according to our literature, but also our proteomic data. And this assay measures the activity of CKB by the conversion of tenofovir monophosphate, which is the precursor for the active form of tenofovir diphosphate. And we can measure the formation of tenofovir diphosphate by mass spectrometry, but specifically by adding substrates for this enzymatic reaction that are specific or enriched in for particular enzymes. So for example, by adding phosphocreatine, this is an essential substrate for the CKB mediated enzymatic conversion of monophosphate to diphosphate, which in our graph on the below is the CKB substrate condition that measures activity of CKB. But by adding additional substrates such as ATP and phosphoenol pyruvate, we can measure the activity of other tenofovir metabolizing enzymes in addition to CKB, such as PKM or phosphate kinase muscle type. And here, similar to our in vitro data that measured the formation of tenofovir diphosphate within blood-brain barrier cells, we were able to measure the activity of CKB across the three blood-brain barrier cell types. But interestingly, astrocytes, which is very similar to the expression profile we observed, there was over a 3,500% significant difference in CKB activity in astrocytes relative to parasites and endothelial cells. However, for parasites and endothelial cells, this CKB enzyme was also active, just significantly less within astrocytes. And then we also observed a lack of significant difference between phosphate donors and the CKB substrate donor condition, which suggests that CKB may be a primary enzyme for metabolizing tenofovir within these blood-brain barrier cell types, because there's no significant increase in tenofovir diphosphate formation when adding substrates such as ATP and phosphoenol pyruvate for other tenofovir activating enzymes. And so, again, this data suggests that these cells are active in the formation of tenofovir, pharmacologically active, but also differentially enzymatically active in the metabolizing enzyme activity. We then wanted to measure the activity of other metabolizing enzymes for both tenofovir and tricytidine. Here we used primary human blood-brain barrier cells again, but used colorimetric activity assays to measure the activity for pyruvate kinase type M, or PKM, as well as thymidine kinase, or TK, which are tenofovir and M-tricytidine metabolizing enzymes respectively. And here we see quite a bit of variability within samples within a cell type, but this data shows that all three blood-brain barrier cells have activity for both pyruvate kinase and thymidine kinase, but we did not see significant differences between blood-brain barrier cell type. We then wanted to consider how metabolizing enzymes and transporters may change within blood-brain barrier cells, but also as a blood-brain barrier as a whole, in the context of both disease and therapeutic intervention, as these metabolizing enzymes and transporters are typically existing in the activity of HIV infected contacts that are also typically in the context and background of antiretroviral therapy administration. And so it's possible that the expression might change as a response of to exposure to HIV, antiretroviral drugs, or a combination of the two. And so to further examine how metabolism and transport may be affected by both HIV and antiretroviral drugs, we use primary human blood-brain barrier cells, endothelial cells, parasites, and astrocytes, and expose these cells to either antiretroviral drugs, including tenofovir and emtricitabine, at concentrations that have been previously used extensively in literature for these types of assays, HIV, or combination of antiretroviral drugs and HIV together, as well as a vehicle condition. And we expose these cells for 24 hours to these different conditions. And we use proteomics by mass spectrometry again to measure the concentrations of different proteins of interest. And here, we're able to measure the concentrations of several tenofovir and emtricitabine metabolizing enzymes and transporters across the variable conditions, so either vehicle, antiretroviral drug exposure, HIV exposure, or a combination of the two. And here, we're able to appreciate that we can measure differential concentrations of transporters, tenofovir and emtricitabine metabolizing enzymes between these conditions, suggesting that antiretroviral drug exposure, HIV, or combination of the two may impact the concentrations and thus the metabolism and transport for tenofovir and emtricitabine. But interestingly, we observed these effects only in astrocytes and parasites. We did not see any significant differences between conditions in endothelial cells, which is important to consider because when considering blood-brain barrier studies, but also how we think about antiretroviral drugs interacting with the blood-brain barrier, it's typically in the context of brain endothelial cells used by literature. And so this shows that we might need to also include astrocytes and parasites in these studies, as they are also important and essential blood-brain barrier cell types. And so we accumulated and summarized this data in this table to look at the different protein changes as a response of exposure. And here we're able to measure metabolizing enzyme changes on the left, emtricitabine metabolizing enzyme changes, as well as transporters for several different proteins, again, only in parasites and astrocytes. And interestingly, we see proteins that respond significantly to all three conditions, so ART, HIV, or combination of the two in terms of exposure, such as adenylate kinase 2 or AK2, which is a tenofovir metabolizing enzyme, particularly within astrocytes for these differences in concentration. We see, for example, proteins such as creatine kinase brain type or CKB in parasites that only respond to one exposure condition, so only concentration changes in response to antiretroviral exposure. And then interestingly, we see proteins that respond only to a combination of both HIV and antiretroviral exposure. In the case of cytidine monophosphatidylglycerate kinase 1 or CMPK1 at the bottom here, which is an emtricitabine metabolizing enzyme, particularly within astrocytes. We see that a combination effect occurs between the addition of or an exposure of both HIV and ART together. And then, of course, we see several transporter changes as well in response to ART, HIV, and HIV and ART together. And so this data together shows that these transporters and metabolizing enzymes for tenofovir and emtricitabine are changing in expression and may be regulated by exposure to HIV, ART, or combination of the two, which is important to consider how we think about metabolism and transport of antiretroviral drugs in the context of disease, specifically HIV, at the blood-brain barrier, but also, of course, exposure to these antiretroviral drugs too. So there could be changes in this metabolism and transport, and thus could impact HIV treatment within the brain and access of antiretroviral drugs to the brain. And so we also wanted to see if there are other proteins in addition to our tenofovir and tricytidine metabolizing enzymes of interest and our transporters of interest. And so we wanted to see if HIV and ART might be changing other pathways within our blood-brain barrier cell types that may explain the changes that we see in our metabolizing enzymes and transporters, as these proteins are not only important for tenofovir metabolism or tenofovir and tricytidine metabolism and transport, but they also play roles in many other pathways in the cell as well. And so to investigate this, we conducted the same experiment where we exposed primary human blood-brain barrier cells to ART, HIV, a combination of the two or vehicle, and then used proteomics by mass spectrometry to quantify our changes in concentration that may be as a result of these exposure conditions. And then we use a web-based software platform called Simplify, which is for proteomics data that essentially analyzes all of the proteins within the sample that may be changing and relates them to pathways using a geoanalysis that to mark pathways that could be changing holistically as a result of different exposure conditions from the different proteins that are changing within that pathway. And so, interestingly, just like our nucleotide metabolizing kinases and our transporters that changed, we saw pathways that were changing, many pathways that are changing, but specifically within astrocytes and parasites. These three graphs show the pathways that are significantly differentially regulated in astrocytes as a response to HIV exposure on the left, ART exposure in the middle, or a combination of HIV and ART on the far right. And there are several pathways here, but for ease of analysis, we categorize the different pathways into 5 different categories to understand holistically the types of pathways that are changing. So here we see pathways that are changing in cell transport and regulation, which makes sense because we saw many transporter proteins that were impacted, particularly transporters that, as I just showed, important for tenofovir and tricytidine transport. We see, of course, significant differences in metabolism, which again makes sense, but also, as I mentioned, several of the tenofovir and tricytidine metabolizing enzymes also play roles in important cell metabolism pathways, such as glycolysis and glucose metabolism, as well as citric acid cycle. And so it's important to consider that the proteins and the metabolizing enzymes that we're interested in may be affected by our exposure conditions to HIV and ART as a result of holistic broad pathway changes. And then we also see pathway changes that were particular to immune response and signaling, which makes logical sense when we expose these cells to HIV, but also antiretroviral drugs, which goes on to say we see significant pathways that are differentially impacted that are related to HIV pathogenesis, as we consider astrocytes can be reservoirs of HIV and are infected by HIV. And then, of course, too, we see several pathways that are impacted by cell-cell communication, which is important to consider that we're thinking about the blood-brain barrier, which is and relies on and built by cell-cell communication between astrocytes, parasites, and endothelial cells. And so this data together, by looking at all these different pathways, holistically is understanding how HIV and ART together may be changing metabolism and transport for tenofovir and tricytidine, but also other pathways that are important for proper cell functionality, which may result in why we see a dysregulated blood-brain barrier in the context of HIV infection, which is reported by literature. And of course, as I mentioned too, we can conduct the same experiment for parasites as well. And we see many pathways, many similar pathways that are impacted by HIV, ART, and HIV and ART exposure. And interestingly, we see less pathway impacts that are related to HIV pathogenesis, although we do see that some of these pathways related to HIV pathogenesis are impacted, as parasites have been observed to be infected by HIV in vitro by literature. But not to the same extent as astrocytes. And then, of course, too, we see differences in cell transport and metabolism and regulation pathways, cell-cell communication, and immune response. So this very similar pathways that we were able to categorize for astrocytes. Interestingly, these same pathways, if we look at what happens to these pathways in endothelial cells after exposure to HIV, ART and HIV and ART together, we don't see any significant differences in pathways that were impacted. And so, again, this data reinforces that these cells, particularly parasites and astrocytes, are dynamically regulated by HIV and antiretroviral drugs, and as well as those two exposure conditions in combination, but also reinforces the fact that we really have to consider parasites and astrocytes as important dynamic players of the blood-brain barrier that can change, particularly in a pharmacological impact for tenofovir and tricytidine metabolism transport, but as I mentioned, for other cell pathways as well. And so together, this data has been able to demonstrate the pharmacological capabilities, specifically the metabolism transport of tenofovir and tricytidine within the blood-brain barrier cells. And I demonstrated this by quantifying the active metabolites as well as the parent forms of tenofovir and tricytidine within blood-brain barrier cells in vitro, as well as within macaque astrocytes and brain tissues in vivo. We are able to quantify concentrations of tenofovir and tricytidine metabolizing enzymes and transporters within blood-brain barrier cells, and found that the concentrations as well as the activities of these proteins are differential between blood-brain barrier cells, which may suggest and explain the heterogeneous measurements of both the active metabolites and parent drugs of tenofovir and tricytidine. And this data together shows that blood-brain barrier cells contain specific activities in the metabolism and transport that may be distinct from the liver biomeasurements within hepatocytes, and may together impact the heterogeneous distribution of antiretroviral drugs, specifically tenofovir and tricytidine within the brain, which we need to consider in the context of both HIV and therapeutic intervention, as we also saw that HIV and ART may differentially impact metabolizing enzymes and transporters, as well as several other important cell pathways, but specifically within astrocytes and parasites and not endothelial cells, which further highlights the importance of considering parasites and astrocytes within literature, particularly within a pharmacological consideration. And so together, all this data, hopefully I've convinced you that we're able to measure heterogeneous tenofovir and tricytidine metabolism and transport at the blood-brain barrier in the context of disease and therapeutic intervention, which is important to consider for antiretroviral treatment for patients who have persistent virus within the brain that results in significant neurologic consequences. And so together, this lovely data could not be without significant help from both my labs, Dr. Deanna Williams at the Emory School of Medicine, as well as Dr. Ben Orsburn at Johns Hopkins. This data was collected with the help of several individuals within the lab, particularly Stephen within the Williams lab and Dr. Colin Eberhard, who presented, I believe this time last year, maybe in the spring of last year, on similar work of tenofovir with CKB. Of course, I want to thank my thesis committee, who has been wonderful in mentorship, the Department of Pharmacology here at Johns Hopkins, and of course, our funding sources for supporting this work. And with that, I will take any questions that you may have.
Okay, thank you so much. If anyone has questions, you can either put them in the chat or you can turn on your camera and your mic and you can ask them directly. Dr. Call, do you want to, Marcus, do you want to ask your question?
Oh, okay, thanks. Thanks, Hannah. Very nice talk. Really, really beautiful work, and I mean, really interesting. I have a lot of questions, and I'm trying to, but let's start. So I mean, so you looked at all these cells separately, right? And just by looking at, I mean, in the blood-brain barrier, of course, they sit all together, right? And they talk to each other. So have you looked at anything, how, some of the profiles that you see, how perhaps this looks when you compare it, to the community that cells talk to each other?
Yes, a very important question. That is our next step. Our lab has a really wonderful co-culture model system that's within a Transwell system, looking at endothelial cells, parasites, and astrocytes all together, and it'd be wonderful to look at those same experiments. But currently within the field, for blood-brain barrier cells, we have a very limited understanding of baseline metabolism and transport, and also the effects of HIV and ART on each cell type individually. And so we wanted to build a foundation, starting ground up, on how these cells may be responding, which we then, of course, want to look at in a cold culture system, which I'm sure these will change to an extent. But without that initial groundwork, it will be very hard for us to say to what extent these might be changing.
Yeah. Oh, no, that makes sense. I mean, you have to start somewhere, right? And it's easier to, with the thing you said. And so I was wondering, is there any idea, or, because, and maybe I missed this in some of the slides that you saw, because, I mean, one of the important functions, right, of astrocytes also is handling of neurotransmitters. And, what people have been looking at often is, for example, glutamate uptake, glutamine conversion to glutamine and transport. Have you seen anything there changing? Or and the other question is, do the astrocytes that actually sit at the blood-brain barrier actually contribute in this function, or are they separate from the astrocytes that are part of the synapse?
Gotcha. So just to make sure I understand your question, are you asking about looking at the important cell functionality of different neurotransmitters?
Yeah, like glutamate, for example, glutamate transport, right?
Yes,
I mean,
in
there's been
response
done a lot of
to,
work on glutamate transporters, right, in astrocytes, right?
of course, yeah, in response to specifically looking at the effects of HIV and ART specifically.
For example, yeah.
Yeah, that's a really important question. Really interestingly, for the pathways that we're able to analyze, we do see some significant differences in pathways related to neurotransmission and glutamate within our astrocytes that we're able to observe in our simplified data. In terms of looking at pathways that I showed that were relative to metabolism, transport, and other types of regulation, the effects were not as significant as the other pathway differences that we observed. But we did see pathways related to transmission across chemical synapses, which is the specific geo term that Simplify highlighted, that were upregulated or significantly different, which was as a result of upregulation, downregulation of proteins within that pathway.
So maybe one last thing.
Of course,
Have you ever tried to combine the drugs? Because also if you think about if they share some of these transporters, or so if you combine, how do you see how, if because it's usually combined antiretroviral treatments, right? Have you looked at this?
Yeah, so the combined antiretroviral drug exposure, so tenofovir and tricytidine in addition to dolutegravir. Yeah, so all of the exposure experiments that we conducted, the antiretroviral exposure was in a combination. So we wanted to make sure it mimics what we'd see in clinic. It'd be really interesting to parse through what was specific to tenofovir versus tricytidine versus dolutegravir. But we first wanted to start looking at the whole regimen in addition. Yeah, and it would be very interesting to look at other important antiretroviral drugs, for example, like efavirenz and rubin and other drugs like that. But yeah, in combination.
Cool. Thank you very much.
Thank you. Takashi, did you have a question?
Sure. I had a very nice talk. I really enjoyed it. So my question is, I guess these products are taken orally. So do you know anything about the composition of the metabolites by the time it hits blood-brain barrier? Like, is predominantly those are already phosphorylated by the time it gets to the blood-brain barrier? Yeah,
that's a good question. The short answer is that we don't know. Understanding the pharmacodynamics is that they shouldn't be, just because the phosphorylated forms of these drugs are very charged and very bulky. And currently we don't know any we don't know any transporters actually that transport the phosphorylated forms, which is important to consider because what we think is happening, right, is that tenofovir and tricytidine are accumulating within our blood-brain barrier cells. And then once they're phosphorylated intracellularly, those metabolites are then trapped within those cells. And so, which is very important for considerations of antiretroviral availability within the brain, right? And so
Right.
it could be that they're influx transporters that are influxing the phosphorylated forms, but currently that is not known within the field. And so that's kind of our limitation there, just structurally from the metabolite formation.
All right. Okay. Thank you.
Yep. I will say too that tenofovir
Mhm.
and emtricitabine, but specifically tenofovir, their plasma protein binding content or percentage is very low. And so they're able, which is a big reason why we're probably seeing it in the brain, is that they're not binding to plasma proteins. And so they're able to be unbound and, of course, cross the blood-brain barrier. Other antiretroviral drugs have quite variable plasma protein binding content.
So when you say cross the blood-brain barrier as a prodrug.
So not prodrug is tenofovir. So the prodrug form of tenofovir is cleaved by esterases when it's taken as an oral drug. So in the gut, it's usually in the the gastrointestinal system. They're by esterases. Yep. So it'd be the tenofovir form. Yep. Let's see. And then I see a question from Carl Goodkin about MMPs and TMPs and their homeostasis with respect to the effects of tenofovir and tricytidine on the integrity of the blood-brain barrier. We have not looked specifically, or I have not looked specifically at those proteins. It'd be very interesting to see. We do see that there are, in the pathway analysis as a response to HIV and to ART, there are changes in proteins that are important for blood-brain barrier integrity and communication. And so with those specific proteins, I have not looked at, but the general pathway that is affected, but definitely would be worth taking a look for sure. Thank you.
Thanks, great presentation.
Thank you. And then Erin, I see a question about are only macaques that are injected intramuscularly and not subcutaneous. That's correct. Yes.
You might want to check with some of the people that did that actual work. I don't think that's correct. Some of the concentrations too, maybe follow up with the primate group here.
Will do.
I think some of those are off by quite a bit in some cases.
Yes, Cecilia.
Thank you, Hannah. It was gorgeous. So my question is, I understand that you added HIV at a 5 nanogram per ml, something like this. So you don't have actually active infection in your system. Do you think that things would change if you introduced, let's say, microglia cells that are infected, or some supernatants, or things that would better mimic the actual situation of an infected brain?
Yeah, absolutely. That's a great question. So that concentration we used that was pulled from literature, from previous studies that have been looked at, particularly with the TAP protein. I absolutely think that the changes that we see in response to higher concentrations of HIV would be, my predictions would be much more drastic. Just because of what we know with, particularly with blood-brain barrier dysfunctionality as a response of it to HIV infection. With microglia as well, it'd be very interesting to consider those as well. We did isolate microglia from our rhesus macaque antiretroviral healthy cohort, and so it'd be very interesting to look and do the same analyses within microglia. We just haven't gotten to those analyses yet, just because we're focusing first on blood-brain barrier cells. But absolutely, I think the changes would be far more drastic, and which is important to consider too, because our estimate, as you mentioned, is a conservative estimate, right? And so if we're seeing significant changes with this conservative estimate, with rapid infection and high concentrations of viral load within the brain, I would imagine there'd be much more drastic changes.
Thank you so much.
Thank you. Any other questions? Wonderful. Well, if there aren't any other questions, I will say thank you very much again for the opportunity to present. This work was recently published in ACS Pharmacology and Translational Science. And so if you have further questions and want to take a look, it is out as of a week and a half ago. But thank you again for the opportunity to share my work and for all your wonderful questions. It's great to present to such a great community. So thank you.
Thanks, Hannah. Really
Thank you.
appreciate it. It was a wonderful talk.
Yeah,
very
thank you
nice.
so much.
Thank you.
Yeah.
Thank you.
Okay, and for
Right.
everyone next week,
Uh-huh.
we'll have Rebecca Easter and Kay Ridgway, and they're presenting facial emotion processing and psychosocial predictors in people with and without HIV living in Uganda. So I'll send
right.
the reminder about that, but that's the talk next week.
All right.
Okay, thanks
All right.
everybody.
Thanks, everyone. Bye-bye.
Thank you.